Hello, my name is Alhajeh, and today I'm presenting about cellular respiration and photosynthesis. Let's start first with cellular respiration. In the formula of cellular respiration, glucose reacts with oxygen, producing carbon dioxide, water, and most importantly, ATP, which is the source of energy for the cell. As we can see in the photo, cellular respiration is broken down into four stages. Stage 1, glycolysis. Stage 2, acetyl-CoA synthesis. Stage 3, citric acid cycle. Stage 4, oxidative phosphorylation. Stage 1, glycolysis. Glycolysis is the process of breaking down glucose into 2-pyruvate. Also, glycolysis is divided into 10 mainly steps and 3 phases. In step 1 of glycolysis, it begins with a molecule called glucose. ATP donates a phosphate group to glucose, creating a glucose 6-phosphate. In step 2, an enzyme rearranges the structure of the substrate without changing the molecular formula, creating fructose 6-phosphate molecule. In step 3, another phosphate group is being added to fructose 6-phosphate, creating fructose 1,6-biphosphate and ATP. Steps 1 through 3 are called phase 1, preparatory phase, with the consumption of 2 ATP. In step 4, fructose 1,6-biphosphate is being divided into two molecules, which are DHAP and GAP. This process is mainly driven by an enzyme. Step 4 is known as phase 2, the cleavage phase, because 6-carbon sugar is being cleaved into 2,3-carbon molecules. In step 5, glyceraldehyde phosphate is being removed, and it will be used in step 6. In step 6, GAP is the only molecule that is being used. Anorganic phosphate is being added to the glycerol hydride 3-phosphate, producing 1,3-biphosphate glycerate. In step 7, phosphate group is being transferred from 1,3-biphosphate glycerate to ADP to form ATP and 3-phosphoglycerate. In step 8, phosphate is being relocated by an enzyme from the third carbon to the second carbon to form 2-phosphoglycerate. In step 9, H2O is being removed from 2-phosphoglycerate to form phosphopyruvate acid, PEP. In step 10, an enzyme transfers a phosphate from the phosphoenolpyruvate PEP to ADP to form pyruvic acid, and ATP results in step 10. 2 ATP net are produced and 4 ATP in total. Stages 1 through 10 are known as the payoff phase, with the production of 4 ATP and 2 NADH. We produce 4 ATP in total while we use 2 ATP during that cycle. And stage 2 of cellular respiration is acetyl-CoA synthesis, where pyruvate is broken down to create more energy. This stage acts as a link between glycolysis to citric acid cycle, and it also takes place inside of the mitochondria. The mitochondria consist of inner membrane and outer membrane. The space between the inner and the outer membrane is called intermembrane space. Space enclosed by the inner membrane is called the mitochondrial matrix. Pyruvate converted to acetyl-CoA synthesis happen in the mitochondrial matrix. First, pyruvate molecule oxidized and splits to form carbon dioxide. Electron lost in the process and are donated to NAD+, which reduce to NADH. Second, the remaining of pyruvate CH3OC is being transferred to coenzyme ECO-A. A molecule carries acetyl group to the next set of reaction. The reactions are catalyzed by an enzyme called dehydrogenase complex. The synthesis of one molecule of acetyl-CoA from pyruvate results in the formation of carbon dioxide and one molecule of NADH. The third stage of cellular respiration is citric acid cycle. The citric acid cycle begins with acetyl coenzyme A combining with the oxalocitric acid in order to create 6-carbon molecule which is known as citrate. Through this cycle, citrate is being reduced and oxidized in a bunch of steps to get back to oxalocitric acid. The most important in this cycle is to notice that there is CO2 are produced or released, NADH converted or reduced to NADH, NAD plus reduced to NADH, ADP is reduced to ATP, FAD is oxidized to be FADH2. Also, these are the inputs for the electron transport chain. That's why they matter in the citric acid cycle. At the end of the citric cycle, the net production is 1 ATP, 3 NADH, and 1 FADH2. Also, I wanted to mention that the citric acid cycle happens in the mitochondrial matrix. The fourth stage of cellular respiration is electron transport chain and oxidative phosphorylation. The electron transport chain consists of 4 complexes, in which uses the products that is created in the citric acid, which are NADH and FADH2. NADH and FADH2 are reduced into NAD and FAD, which the electrons that is being reduced are used as an energy in order to go through the complexes. As we see in this picture, the electrons are pumped from complex 1 to complex 2 through the help of the coenzyme Q transport electron from complex 1 to complex 2. Also, from complex 2 to complex 3, we have also CoQ and CoQH2 enzymes are helping to pump these electrons. Also, from complex 3 to complex 4, the cytochrome C, the enzyme, is helping to pump the electrons by producing O2, enough O2 and H pluses in order to produce H2O. This picture explains proton transport and ATP synthesis. This picture shows that protons are going from high concentration to a low concentration. Also, it shows that after the protons are being in a low concentration, they're going to ATP synthase. ATP synthase use electrochemical proton gradient to drive the synthesis of ATP. Photosynthesis. Photosynthesis is a process in which plant cells use in order to produce ATP using carbon dioxide and carbohydrates. The type of reaction that is used in photosynthesis is a redox reaction where CO2 molecules are being reduced to a higher energy carbohydrates, or as known sugar, as is shown in the reaction formula. Also, H2O is being oxidized in order to form oxygen. The process of photosynthesis happens in different parts of this chloroplast. But first, let's look at both photosystem 1 and 2. Photosystem 1 and 2 take place in the thylakoid of the chloroplast. During the photosystem, one absorb of the light transfer to molecule as electron acceptor. Reaction center becomes oxidized and the electron acceptor is reduced, resulting of the conversion of light to chemical formula. The two photosystems provide enough energy to pull an electron from water and then use it to reduce NADP. The photosynthetic electron transport chain connects the two photosystems. Photosystem 2 provides electrons to the beginning of the electron transport chain. When photosystem 2 loses an electron, it is able to take another electron from water or H2O. Photosystem 1 energizes the electron with a second input of light energy, so they have enough energy to reduce NADP+. Photosystem 1 is not as strong oxidant to split water, and photosystem 2 cannot produce electron with enough energy from NADPH. Electrons passes between photosystem 2 and 1 through cytochrome B6F complex. Plastoquinone carries electrons from photosystem 2 to the cytochrome B6F complex, while plastocinin carries electrons from cytochrome B6F complex to photosystem 1 by diffusing through the thylakoid lumen. The oxidation of water releases protons and O2 into the lumen. Then cytochrome B6F complex functions as a proton pump as electron transit through complex. Some of the energy that is released by redox reaction is used to drive protons from the stroma side of the thylakoid membrane to the lumen. When protons pass through the enzyme ATP synthase, ATP is generated. Due to the variation in light intensity throughout the day, photosystem work in a cyclic manner. When light levels are low, light is shared evenly between the two photosystems. However, as light levels increase, the light energy absorbed begins to overwhelm the Calvin cycle's ability to make use of NADPH. When no NADP plus is returned from Calvin cycle, the high energy electrons can damage the cell. To keep this from happening, electrons are shunted into an alternate pathway that increases the production of ATP while decreasing the production of NADPH. Electrons from photosystem 1 are redirected from ferredoxin back into electron transport chain. These electrons re-enter the electron transport chain upstream of the cytochrome B6F proton pump. The Calvin cycle consists of three stages: carboxylation, reduction, and regeneration. Carboxylation is where happened the addition of a CO2 producing 3-phosphoglycerate 3-PGA. And reduction, NADPH transfer a high energy electrons. NADPH is changed to NADP+. And regeneration, ATP is transferred to ADP. This reaction is driven by an enzyme called ribisco. As we move to photorespiration, we find that ribisco adds O2 instead of CO2 to RUBP, resulting in one molecule with the 3 carbon atoms, 3 PGA, and one molecule with only 2 carbon atoms, 2 phosphoglycate. This is the only difference that is being compared to Calvin cycle. Thank you. I hope you enjoyed my explanation of photosynthesis and cellular respiration. Two fun facts before we end up our video. Did you know that you could perform cellular respiration without an oxygen? which is called fermentation. Also, did you know that photosynthesis and cellular respiration are the opposite reactions?